Hello, welcome students. Today we are going to start a new chapter from the Lippincott, chapter number 8, and the title of today is the Introduction to Metabolism and the Glycolysis. Metabolism, as we all know, that it is called as the sum of all the reactions taking place in a living organism. This is the sum of all the reactions, sum of all the biochemical reactions, all the biochemical reactions which are taking place in living organisms. So in the cells, the metabolic reaction rarely occur in the isolation. Instead, that they are organized into multiple sequences and called as pathways. In the living bodies, no reaction occur in isolation, but it occurs in a series manner and in a sequential enzymes. These serial reactions are also known as pathways. In the pathway such as glycolysis, the product of one reaction serve as the substrate and the subsequent reaction. Most pathways are classified as catabolic. Now we all know that metabolism is categorized into two categories. One is known as catabolic. Catabolic reactions are also known as degradative reactions in which complex organic molecules are converted into simpler or simple molecules. And when we discuss about anabolic reactions are also known as synthetic reactions in which simpler compounds combine to form complex molecules. Catabolic pathways break down complex molecules such as we have already studied in the catabolic reaction, the complex molecules such as proteins, carbohydrates, and lipids are converted into simpler compounds such as carbon dioxide, amino acid, water. So these are known as the catabolic reactions. So when we talk about anabolic reaction, we all know that the simpler molecules combine to form the complex one. It is known as the anabolic pathways or the synthetic pathways. Uh, Very simple example, as the conversion of the glucose, alpha glucose, simple molecule that is converted into glycogen. It is a polysaccharide and glucose is a monosaccharide. Is a simple example of the anabolic reactions. Different pathways or the different reactions also intersect with each other. For example, there are different reactions that take place simultaneously. For example, if we know about that, the glycolysis, in which glucose is converted into two molecules of pyruvic acid. At the same time, this pyruvic acid is also attached with the oxidation of the pyruvic acid, and oxidation of the pyruvic acid is attached with the Krebs cycle. Similarly, fats is also converting into fatty acids and glycerol, and these are also interconnected with each other. So all the reactions making the mesh or a chain within the body, so these reactions intersect with each other. So at the end, the metabolism is the sum of all the chemical changes occurring in the cell, a tissue or a body, and the metabolites are the intermediaries product of the metabolism. So students, as we have already discussed it, that within the body, within the cell, no reaction takes place in isolation. There is the involvement of the multi-enzyme sequences. So the product of one reaction may be the substrate for another enzyme. So this in this way the reaction takes place in a series of reactions. And these multi-enzyme involvement has very purposeful movement of the metabolites and depicting the effects on the flow of the intermediates. That means the formation of the product in a reaction may have an effect on the regulatory function of the reaction. It may enhance, it may inhibit, it may reduce the reaction. So one product may be act on the reaction in a positive or a negative way. So we have discussed that there are two types of reaction, catabolic and the anabolic reactions, and the metabolic pathway is in that type of reactions in which multi-enzyme systems are involved, and the products which are formed during these reactions are known as intermediaries or the metabolites, and each metabolite can have the catalytic or the regulatory effect on all the reactions, which we will see in the future in the next slides. For example, by a drug or an inherited deficiency of enzyme, so we can regulate these enzymes by administering a drug which can inhibit a certain enzyme, or it can stop or enhance the particular efficiency of the enzyme. Or if we will see that if a person have the inherited deficiency of any enzyme, how this enzyme will have the bodily effect on them, the positive or the negative way. So all our topic today represents that how this will work. Here you can see that different pathways or the sequential enzymes involved in our body. The main streamline is in the middle. It is representing the glycolysis, the formation of the acetyl coenzyme A and the TCA cycle, and the side reactions. And that means the fructose 1-phosphate formation. Similarly, the fatty acid, the lipid decomposition, lipid formation, and all the reactions, as you can see that they are integrated with each other. So in the chapter we will see it in the detail in the later slides. In the catabolic pathways, the main purpose of the catabolic pathways in the cell is to capture the chemical energy, and this energy is available to the body or a cell or a system in the form of ATP, which comes from the degradation or a catabolic pathway of a molecule, and the molecule must be the energy rich molecule. Energy generated by the degradation of the molecule occurs in 3 steps. Here students, you can see the degradation or a catabolic reaction take place in the 3 stages. The step 1. In the step 1, the complex molecules such as proteins, carbohydrates and fats, they are converted into or degraded into its simple monomers. Suppose protein is converted into amino acid, carbohydrate will be converted into monosaccharides, and fats will be converted into glycerol and the fatty acids. All these amino acid and monosaccharide and the glycerol, they are formed in the stage 2. Now they are again converted into by some chemical reactions, which we will see later on in the next slides, into acetyl coenzyme A, and ultimately this acetyl coenzyme A will enter into a TCA cycle or a Krebs cycle. The main purpose of the degradation of these complex molecule into simpler one, that we gain energy step by step in each reaction, so we can gain energy in the form of ATP. So this is the main purpose. So here we have 3 stages. In the first stage, complex molecules, protein, carb, it will be converted into amino acid, monosaccharide and glycerol. Then these monomers will be converted into acetyl coenzyme A. Then this acetyl coenzyme will enter into the Krebs cycle, and then oxidative phosphorylation, as you have read in the earlier classes. So we can obtain the full benefit of the energy rich molecules in the form of ATP. Here the first step is explained again. First of all, hydrolysis of the complex molecules. In the first step, the complex molecules are broken down into their components, building blocks. For example, proteins degraded into amino acids, polysaccharides to monosaccharide, and fats, fatty acid and glycerol. So here is the first conversion, hydrolysis of the complex molecule, protein into amino acid, carbohydrate into monosaccharides, and the fats into glycerol and the fatty acid. In the catabolic pathway, the second step, the conversion of the building blocks into simple one, as I have already discussed it with you, that it will be converted into ultimately into acetyl coenzyme A and a few other molecules. Some energies captured as ATP, some energies, because at this level we haven't obtained full form of the energy yet. So after conversion of the acetyl coenzyme A, it will enter into a TCA cycle, so we can obtain maximum amount of energy from that particular molecule. But the amount is small with the energy produced during the third stage of the catabolism. So here is the slide again, the same slide again, that in the case of the tricarboxylic acid is the final common pathway in the oxidation of fuel molecules that produce acetyl coenzyme A. Acetyl coenzyme A generate large amount of ATP via oxidative phosphorylation during electron flow from NADH and flavin adenine, and we all know that the energy can be obtained from NADH and FADH too by in electron transport chain. So the main purpose of breaking the molecule or catabolic reaction is to utilize the energy which is present or hidden in the complex organic molecule. So we can obtain energy from. Before discussing the catabolic pathways in detail, we will first examine the exergonic reaction and the exothermic reaction, because at different events the book has used these two terminologies. And first let's clarify these two terminologies. By exothermic reactions definition, reaction that heat that releases free energy, the free energy for the utilization of for the cell especially. And by exothermic reaction it is represented by delta H, and that energy which releases as the heat from the system. Energy types, delta G is also known as Gibbs energy, and it is known as exothermic, that is known as thermal energy. Delta G is lesser than O, that means energy is producing by the system, and delta H is lesser than H0, means that energy is releasing in the surrounding. Main focus of the exergonic reactions that the energy available to do biological work, and in this exothermic the heat releases in the surroundings from the system. Reaction to temperature not necessarily increases temperature, and it increases the temperature of the surroundings. So main difference between these two, that exergonic reaction do not mean that it does also increase the temperature of the surroundings, but in exothermic reaction the surroundings temperature will also increase. Biological systems may, and the biological importance, it is very high, and it helps in thermoregulation as we have already learned from homeostasis. ATP involvement usually produces or uses ATP, and it usually not related to the ATP production. Both can occur simultaneously, yes, and the production of the exergonic reaction ATP in conversion into ADP, and energy is released, and this energy is available for the biological systems such as muscle contraction, transport, and in the thermoregulation, shivering, and in the sweating, exothermic reaction, radiations outside the body to reduce the bodily heat. Cellular respiration, it is exergonic ATP generation, and it is also exothermic heat production. Purpose is the body perform work, movement, senses, and transport, and it is thermoregulation, that means the maintenance of body temperature. Now the difference between NADH and NADPH, both looks very similar, but their role in the body or a cell is very different. First of all, it's a full abbreviation. By full abbreviation, NADH means nicotinamide adenine dinucleotide, and it is a reduced form because hydrogen is present with it. And the second one, NADPH, nicotinamide adenine dinucleotide phosphate, it is also a reduced form. Main role, its production is involved with mainly with the reaction of catabolism, as we saw it in many instances, that in the glycolysis is a catabolic pathway. It is formed NADH, and it is a biosynthetic and protection pathway. Anabolic reactions. Type of reactions, oxidation reactions, and NADPH is used in the reduction reactions or synthetic pathways. Primary function, it produces ATP in the electron transport chain, biosynthetic reactions and antioxidant defense. Location in the cell, NADH is produced mainly in the mitochondria, but the NADPH is produced in the cytosol. Generated it, as you already know, that glycolysis, pyruvate, dehydrogenase and Krebs cycle, and it is formed during the pentose phosphate pathway used in oxidative phosphorylation, and it is produced during a fatty acid synthesis and cholesterol synthesis, which we will learn in the later chapters. Energy production equal to almost 2.5 ATP or 3 ATP, does not directly produce ATP. It is used in the catabolic reaction and it is involved in the anabolic reaction. Antioxidant role, it has no direct role in the antioxidant. It maintains reduced glutathione to combat oxidative stress. Human example, NADH from Krebs cycle enter electron transport chain, as you have already know. NADPH used in the liver for fatty acid synthesis and in RBC to prevent oxidative damage. Now again, anabolic pathway. Anabolic pathway is also known as divergent process because many small molecules, simple molecules, organic molecules such as amino acid, they join together to form a wide variety of proteins. So it is diverging. So it produces many types of different organic compounds. So it is known as the divergent process as compared to the catabolism, which is the convergent process. Anabolic reactions require energy, as we already know, and also known as endergonic reaction, which is generally provided by the hydrolysis of adenosine triphosphate and inorganic phosphate. Catabolic reaction generate energy and are exergonic. Anabolic reaction often involved chemical reductions in which the reducing power is mostly provided by NADPH or phosphorylated NADH. So these are the metabolism. Metabolic reactions, we all know that metabolism is known as the sum of all the reactions taking place in living systems are known as the metabolism. The metabolism is categorized into two categories. One is the catabolism and other one is the anabolic reaction. Catabolic reactions are also known as convergent processes because it converges into a few types of the compounds, while the anabolic reactions are those which are known as the divergent processes because different simple monomers combine to form various types of proteins, polysaccharides, lipids and nucleotides, because these reaction is diverging and producing many types of products from the simple organic compounds or monomers. So these are known as the divergent processes. All the metabolic reactions are well regulated. They are well coordinated to each other. Simply, it is sometime turn off and turn off because according to the needs of the cell. So sometime these reactions can be turned on and can be turned off. Number two, individual cell function as a part of community of interaction among tissues, not in isolation. Simply that the different cells which are present in the body, they are well coordinated with each other and they observe keenly about the requirement of the body. So they act as a helper or a community process. So when we want to turn on a gene, one cell or within the cell, a signal is generated and the cell can activate its transcription or its translation, or if there is no need of that product, we can turn off that gene. This sophisticated system have evolved in coordinated functions of the body. Now the regulatory signals are of many types. These can be the hormones, these can be the neurotransmitters via neurons, and these can be the nutrients availability. If some certain kind of nutrient is available, or if it is not available, it can also inhibit enzyme action, or it can promote the enzyme action to enhance or to inhibit the cell function in a very well coordinated manner. So all the reactions are well regulated according to the needs of the body. So there are two types of metabolic regulation. First of all, the intracellular communication. The signals which generate within the cell is known as intracellular communication. For example, from point A, the rate of metabolic pathway can respond regulatory signals that arise within the cell. Number two, these regulatory signals can be in the shape of substrates, in the shape of product inhibition, alteration in the levels of allosteric activators or inhibitors. These we will discuss in later on. For example, a substrate is present in more quantity in the cell. It can activate a certain type of cell, or its enzyme to conduct a chemical biochemical reaction, or it can be a product can inhibit by feedback mechanism a cell, or its enzyme, or its quantity. If the product is present in a higher amount, so its quantity alters the enzyme structure in such a way that it can promote or even increase the enzyme activity. These intracellular signals elicit rapid response. The other advantage of the intracellular communication, because all the stimulus or a signals is producing within the cell, so intracellular communication is faster as compared to the intercellular communication. The second type is the intercellular communication or between the cells. It is necessary for the survival of the organism because all the cells communicate with each other by different means, and it is essential for homeostasis and for the survival of the organism. The second difference between intra and the inter is that by intercellular communication, mostly or likely, it affects on the gene expression change. So in intracellular, mostly the enzyme inhibition or enzyme activation is going to be done, but in the intercellular communication, gene expression, a gene can be turned on or off by receiving a signal from the extracellular domain. Communication between the cells. There are many types of communications between the cells. These can be the surface to surface contact. You all know by the receptor sites, gap junctions which are present in the cells of the heart for the action potential to develop, allowing direct communication between the cytoplasm of the adjacent cell. That means by the gap junctions or the intercalated discs. Here is a simple example of the intercellular communication. So first of all, this is the sending cell. Sending cell means that this cell is giving a signal in the form of a chemical. It can be a neurotransmitter, it can be a hormone, it is also known as chemical signal or a ligand. This ligand is received by a receptor site. This receptor site is present on the cell membrane, usually present on the cell membrane of the another cell, and there is a series of intracellular changes which results into the response by a cell. A gene can be uh, turned on, or by receiving signal a gene can be turned off. So if there is no ligand, so that the target cell will not respond. So this is a simple example of the intercellular communication. Now, very important by examination point of view, G protein linked receptors and second messenger system. So we have already discussed that in intercellular communication, it can be done by the hormones and it can be neurotransmitters, and we can call them a ligand or a chemical signal. And these ligands or chemical signals are received by the detectors or a receptor site or receptors which is usually present at the surface of the cell membrane. And what are the receptors? Receptors are the proteins often found embedded in the plasma membrane of their target cell. They respond to the ligand by bound to them. Obviously, the ligands will attach with the receptor site and it will take a series of reaction within the cell. It will initiate a series of reaction within the cell, and we called it these the intracellular responses. Many receptors that regulate metabolism are linked to intracellular GDP binding proteins known as G protein, also known as G protein coupled receptors. Now we will look at it in detail how G protein coupled receptors actually works in the cell. A simple animation which I have already told you, that this is the chemical signal known as ligand. This is the receptor site. This receptor site is embedded in the cell membrane, and after attaching the ligand to a receptor site, it will initiate a series of reactions and produce secondary messages which then go into the nucleus, and after entering into the nucleus, it will it will join with the segment of a gene already present in the nucleus. It can turn on the segment of the gene, and it results in the transcription. And after formation of messenger RNA, this RNA will goes out into the cytoplasm again, where the phenomena of translation will take place. This reaction not only increase the transcription, but these type of signals can also turn off the genes, or you can say it can turn off the transcription, or it can turn off the translation, but according to the ligand, but according to the signal what we want from the cell to do. So some ligands, some chemical signals can activate a cell process of transcription and translation, or some signals can inhibit it. So G protein system, how it works. G proteins regulates the production of molecules referred to as secondary messenger, which also named because they intervene between the original extracellular messenger and the ultimate intracellular effect. Second messenger part of the cascade of event that converts ligand or a chemical signal to a response. First of all, we have to understand that G proteins are linked from the outside signal which came from the other cell in the form of a ligand, and it always attach with the receptor site, and it produce a secondary messenger. The secondary messenger system is regulated by two systems again. One is known as phospholipase C system, in which calcium is involved. But now we are looking at the most important part of the other system, that is phosphatidylinositol system, and the adenylate cyclase or adenylate cyclase system, which are particularly important in regulating the pathway of intermediate metabolism. So before we discuss the first one, we firstly describe the adenylate cyclase or adenylate cyclase system, how it produces the secondary messenger. Both the systems are initiated by binding of the hormone ligand. As we have already discussed it, that these type of system mostly involved in the hormone attaching with the receptor sites, such as epinephrine or glucagon. They will attach with the extracellular domain of the GPCR and of the target cell that will respond to the hormone, and now we will see that how they will respond after attaching with the outer cellular GPCR. So this is the GPCR example. Here you can see the plasma membrane, and here you can see the GPCR. GPCR is characterized by extracellular ligand domain. This is the extracellular ligand domain where a chemical signal attach with it, and the 7 transmembrane alpha helices 1, 2, 3, 4, 5, 6 and 7. Now these are the 7 transmembrane alpha helices 1, 2, and these are the 3, 4, 5, 6 and 7. Now it is that intracellular GPCR domain because it is present within the cell. Now this is the G protein which is present inside the cell membrane. It is known as G protein because it has 3 domains. One is the alpha domain, other one is the gamma domain, and third one is the beta domain. To calling it a G protein because it is always attached to a guanine diphosphate, or it can also attach with the GTP, guanine protein, guanine triphosphate. So that's why it is also known as G alpha. So G alpha is attached in its active form in the GTP, and when it is inactive it is attached with the GDP. So when activated, or a signal comes outside and attached to the extracellular domain of the GPCR, a signal is sent via the 7 transmembrane alpha helices to the inside, and the intracellular changes occur and results in the activation of the G alpha. G alpha now has some intracellular changes, modificational changes. Now it is attached with the GTP because in the inactive stage it is attached with the GDP. So when GTP is attached, the GDP is removed. So in the first step, it will detach from the other subunits of beta and the gamma. Now G alpha is now it is in separate form, or it is in activated form, but in the inactivated form it always attached with the 3 subunits are already together, G alpha, G gamma and G beta. But when it is attached with the GTP, it got separated from beta and the gamma. So here, if the hormone is say insulin, another key regulator of metabolism does not signal via GPCR, but instead via receptor that link with tyrosine kinase activity. So in the case of glucagon, for example, the mechanism which will be involved in here is via the GPCR, but when insulin is present as a hormone, it will take another pathway which we will discuss later on by tyrosine kinase activity and not by the GPCR. So firstly we will see that how adenylyl cyclase is activated, how it is formed. Firstly, after the activation of the G alpha protein, G protein, it will activate the adenylate cyclase which is embedded in the cell membrane. Here is the ATP. Firstly, after activation by G protein, which is now activated by the attachment with the GTP, firstly it removes the 2 phosphate groups, and now it will be converted into cyclic AMP. Now because why it is called as cyclic AMP, because there is one phosphate group remaining, and the group is attached with the 5 carbon of the pentose sugar and also with the 3 carbon of the pentose sugar. So now it is become a cyclic AMP which act as a message, intracellular message. Now extracellular message is converted into an intracellular message. Why? Because the conversion of the ATP into cyclic AMP. But when we want to discontinue the process or inactivate it, there is a domain, inheritedly domain present in the G protein, which we have already seen that the CAMP phosphodiesterase. It is the built-in process in the G protein, and after a while, or in the absence of the signal, it also acts as the phosphodiesterase, and it converts the cyclic AMP again by breaking the bond into 5-AMP. Now it is not a signal, or you can say the signal has been removed. Now it is in inactive form. So when it is converted into a cyclic AMP, it is a signal, it acts as a signal. But when that G domain, it convert the cyclic AMP into a 5-adenosine monophosphate, the effect of it is neutralized, and now it becomes a 5-AMP, and the response will stop immediately. Now this is the full story of the mechanism. Now, first of all, the ligand, or a chemical messenger, attach with the receptor site, which is present on the cell membrane, also known as GPCR. And now, because of the attachment of the ligand with the receptor site, intracellular changes occurs, and there are three. This is the G protein, which is present intracellularly. Now you can see that alpha is present or attached with the GDP. Now it is inactive. But due to intracellular changes because of the ligand, GDP is hydrolyzed into GDP. Now the GTP will attach with the alpha subunit of the G protein. Now the alpha subunit will detach from the gamma and the beta subunits and will travel towards the another embedded protein, which act as an enzyme, adenylase cyclase. And what it will do, it will convert ATP into a cyclic AMP, as I have already described to you. Now it will be a secondary messenger. It will act, or you can say now the extracellular signal now convert into a intracellular signal. Now it will again active another enzyme, which is also known as pKa, which we will see later on. And what pKa will do, it phosphorylate the target protein, or keep addition of a phosphate group to a target protein, and now this target protein will become a cellular response, and the cell will do the thing which the signal is made for. So all the signals are well coordinated. So we can read from here, the effect of the activated coupled G on secondary messenger formation is mediated by specialized heteromeric G protein alpha, beta and gamma subunits found on the inner surface of the plasma membrane. G proteins are named because their alpha subunits bind GDP when activated. Okay, it is, and in inactive form it is bound to GDP. So here we can see it can be corrected. It is when it is active, it is attached with the GTP, or when it is inactivated, it is attached with the GTP. So let's take a quick review again. First of all, a ligand. First of all, a ligand, or extracellular signal. Number one, attach with the receptor site. Receptor site is embedded in the plasma membrane. Here is the three subunits, alpha, gamma and beta of the G protein. In inactive form, it is attached with the GTP. But now, because ligand is attached with the receptor site, the GDP is converted into the GTP. Now this GTP is attached with the alpha subunit, and after attaching with the alpha subunit will detach itself from the gamma and the beta and travel towards another enzyme which is embedded in the plasma membrane. Its name is adenylase cyclase. Adenylase cyclase, after attaching the activated alpha, and it will transform ATP into a cyclic AMP. Cyclic AMP in turn activate pKa, protein kinase A enzyme. This protein kinase enzyme phosphorylate or add some phosphate to a already present protein into the within the cell, and this target protein, when phosphorylated, it will act as a cellular response. We will see that in the future that how this cellular response in the next slide, how this cellular response will work. So the ligand binding causes a conformational change in the receptor, triggering replacement of this GTP with the GTP. The GTP bound form the alpha subunit, dissociate from the beta and the gamma subunits, and moves to the membrane bound adenylase cyclase enzyme affecting the. So overall stories we are telling here that first of all, here is the ligand, and ligand attached with the receptor site, and this is the G protein in its trimeric form, alpha, gamma and beta. When G protein is inactivated, it is attached with the guanine diphosphate. But when it is attached with the ligand, so now there is a intracellular change in the receptor site. The conformational change will convert GDP into GTP, guanine triphosphate. The guanine triphosphate will attach with the alpha subunit of the G protein. Now the alpha GTP will be dissociate from the other subunits of gamma and the beta subunit. It will travel and attach to the adenylate cyclase, which is a transmembrane enzyme, and after attaching, adenylate cyclase will be active and it will convert ATP into cyclic AMP. Cyclic AMP is our second messenger. It will active a protein kinase A enzyme which attach phosphate to a already present protein in the cell, and now this protein is phosphorylated and the enzyme is activated and the cellular response will occur. Here again, the many molecules of the active G proteins are formed by one activated receptor. Note the ability of a hormone of a neurotransmitter to stimulate or inhibit adenylate cyclase depend on the type of the alpha protein that is linked. One is the GS, stimulates, and other one is the GI, which inhibits. Now here is the question new rises that the ligand which attaches with the receptor site. Now which type of receptor site it is attached with? We all know that these are the receptor sites which are also known as GPCR. It is of many types. One is known as GS, or other one is known as GI. If the receptor is the GS, it has the stimulatory effect on the adenylate cyclase and it results in the activation of CAMP. But if the GPCR is the GI type, it will inhibit the reaction and it will reduce the production of adenylate cyclase or AMP. So it depends which type of ligand or which type of receptor that chemical messenger is attaching. If the GPCR is of GS type, it will stimulate adenylate cyclase, whereas GI receptor type inhibits adenylate cyclase. Here is another example. This is the GPCR. Now GPCR is S type. So when a ligand is attached, suppose this is the ligand attached to this site, now it will increase the cyclic AMP by the process I have already told you. And if the ligand is attached to the GI side, it will inactivate the adenylate cyclase and the cyclic AMP is reduced or stopped, so it resulting in the inactivation of the stimulus. So we can read from here also Many molecules of the active G proteins are formed by one activated receptor. Note the ability of a hormone of a neurotransmitter to stimulate or inhibit adenylate cyclase depend on the type of G alpha protein that is linked to the receptor. One type designated as GS and stimulates adenylates, whereas GI inhibits adenylate. You can see here, this is the G alpha S, and this is the G alpha I, and I will inhibit the cyclic AMP. Now again, the story has been repeated. The activated adenylate cyclase converts adenosine triphosphate to cyclic AMP. Cyclic AMP activates the serine threonine protein kinase. As I have already told you that after the formation of the cyclic AMP activates protein kinase A enzyme, which is also known as serine threonine, because it activates or attach phosphate group to the threonine or a serine group. So now the inactive pKa will be converted into active pKa. GTP complex are short-lived. Now this is the GTP complex. GTP is short-lived and it is hydrolyzed back into the GDP, and you also know that the GDP is an inactivated form. This causes inactivation of the alpha GPP, its dissociation from the, now it will be dissociate from the adenylate cyclase because it has an inherent quality of phosphatases. So it removes the phosphate group and it convert into GDP, guanine diphosphate, and it will reattach with the remaining of its two friends, gamma and the beta, and now it become inactivated and the signal will be stopped. Now here the activation of CAMP dependent protein kinase A. This is the protein kinase, is a tetrameric, 4 subunits. Two of them is the regulatory units and two of them are the catalytic subunits. This is the cyclic AMP. When cyclic AMP attach with the pKa, the catalytic subunit will dissociate from the regulatory subunit and it now become active. Now after activation, it will attach with the protein substrate which is already present in the cell, and it will phosphorylate kinase activity. It converts ADP into ADP or adenosine diphosphate, and when this protein is phosphorylated, it will become a signal again, and it has the intracellular effect, or it can stimulate the cell or it can inhibit cell functioning. And if in the absence of cyclic AMP, the reverse reaction will occur, and when the phosphate is removed or a protein phosphatase activity act, the protein or a substrate protein will be dephosphorylated, and now it will be no more a intracellular signal, or you can say that the signal has been removed after D. So after attaching with the phosphate group, it will become a phosphorylated protein. Now it become a signal and it has some intracellular response, but when we remove it, because there is no extracellular signal, so there is no cyclic AMP, now phosphorylated protein will be in which the dephosphorylation will occur because of protein phosphatase. Now it will be dephosphorylated protein. Now it will not more no more longer a signal, and the cell will not respond now. The phosphorylated protein may act directly on the cell's ion channel, or if enzymes may become activated or inhibited. As we have already discussed that phosphorylated protein has some has effect on the cell, so it may activate or inhibit some activity of the cell. Not all the types of protein kinases are cyclic AMP dependent. protein kinase C activated in response to phospholipase C signaling. We have already discussed there are two types of signaling system. One is the phospholipase C signaling system, which is not cyclic AMP dependent. It is the calcium dependent, which we will see in the later chapters. So here is the inactivation. The phosphate groups added to the protein by the protein kinase A removed by phosphoprotein phosphatases. Here is the protein phosphatases. Ester action of the phosphatase ensured that the change in the protein activity induced by the phosphorylation are not permanent. So the phosphorylated protein here is not permanent because in the presence of the signal it is phosphorylated and it's permanently become an intracellular effect. But after dephosphorylation, because of protein phosphatase, it will become again ineffective, or you can say the signal has been removed and cell has no intracellular effect right now. We have already discussed initially that cyclic AMP is hydrolyzed to. In the initial slides, we have discussed the cyclic AMP when there is no signal or it is a time dependent. The cyclic AMP is automatically, after a natural biological cycle, it converts into 5-AMP by cyclic AMP phosphodiesterase that cleaves the cyclic 3,5 phosphodiester bond. 5'-AMP is not an intracellular signal molecule, therefore the effect of the neurotransmitter or a hormone mediated increases of cyclic AMP are terminated if the extracellular is known. So cut the long story short, in the absence of the extracellular signal, the cyclic AMP will be converted into 5-AMP, and the 5-AMP is not an intracellular signal right now, and the signal has been terminated. Here is the activation of CAMP and the inactivation of CAMP. So here is that ATP, adenylate cyclase, present in the embedded in the cell membrane. After the removal of phosphate, dephosphorylation, it attach with this phosphate group. One phosphate group will attach with the pentose sugar with the 5' and the 3', so it is also known as 3', 5' AMP. When the signal has been removed, or it has also a time bounded naturally, so when CAMP phosphodiesterase, it also acts as a CAMP phosphorydiesterase, because the adenylate cyclase can also remove the phosphate or break the bond, and it will convert into 5' AMP. Now it is no longer a intracellular signal. In the last, there is a box in the book. The CAMP phosphodiesterase is inhibited by caffeine, a methylxanthine derivative. When you take caffeine, the phosphodiesterase action of CAMP will be inhibited, so it will remain a cyclic AMP. They inhibit phosphodiesterase or a competitive inhibited, so cyclic AMP remains high. Obviously, when caffeine will inactivate the phosphodiesterase, CAMP persists. So in this way, the heartbeat will become, so the signal will be generating continuously, the heart will become raised, and the brain alternate is also increased. Thank you very much.